Okay, hello everyone. My name's Nicholas. I'm just presenting the research from my honours project titled Could Spillover of the Honeybee Saccharide Virus Harm the Native Bee Tetragonula Carbonaria? I'd just like to begin by acknowledging the Kaurna people as the ongoing custodians of the Adelaide region where this research took place, and recognise that their cultural and spiritual beliefs are still as important to the living Kaurna people today. I pay my respects to elders past, present and emerging, and extend that respect to all Indigenous people here today. So just beginning with a quick background on bees. Of course, they're very important pollinators of flowering plants. They're specialized in this role because they consume pollen and nectar as their pretty much their entire diet. The importance of these pollination services to ecosystems and to humans is highlighted by this statistic here about the global worth of pollination and honey production, which is around $20 billion per year, with the pollination services providing the vast majority of that worth. On the left here is an image of a European honeybee, and then on the right, much smaller native stingless bee, Tetragonula carbonaria. So unfortunately, bees face quite a broad range of threats. Chief among these is poor nutrition due to a lack of adequate floral resources, particularly for some of the more specialized native bees. The correct floral resources are not always available because of habitat loss, and of course climate change, both of which are heavily driven by human practices. Additionally, pathogens and parasites and pesticide exposure are significant threats to bees. Our diversity of native bee species is also significantly threatened by invasive European honeybees in Australia, which compete with native bees for access to those floral resources. It's also becoming apparent that there's the potential for some of the pathogens of European honeybees to be, for native bees to be susceptible to those pathogens as well. And this is the focus of my research. So of these honeybee pathogens, viruses are one of the significant groups. The honeybees being a social species and a widely managed species are harbour a broad range of viruses, and being a communicable disease, they persist in those populations. Most of these viruses are RNA viruses, and like all viruses, they replicate in live hosts and spread from individual to individual within the colony. Some of the most significant of those RNA viruses are deformed wing virus, black queen cell virus, and of course saccharide virus, which is the focus of this study. So it's understood that there are a number of factors influencing this interspecies virus transmission that's suspected between honeybees and native bees. The first of these is co-foraging between managed bees and wild native bees. So this is the direct, the site of where that spillover takes place when a managed bee, which has the virus on it, visits a flower, deposits the viral material, and a native bee picks it up. And that's the avenue by which a novel pathogen can establish in a new host. The close genetic relationship between these species is also thought to play a role, and interacting effects from other threats. So native bees already with weakened immune systems from pesticides or being poorly nourished are thought to create an avenue whereby a novel pathogen, a novel virus can establish an infection in that population. So what is known currently in native Australian bees research has shown that deformed wing virus, which is a virus not present in Australia, was capable of replicating in genera from two different in genera of two different native Australian bees. This research was conducted overseas, obviously, so as not to introduce that virus here. But closer to home, studies in Australia have found that sapwood virus is present in a range of native bee species, including our native stingless bees. But as of yet, no one has investigated whether sapwood virus is capable of causing pathogenic effects in this species or any other species, or whether it's capable of replicating, which is a definite prerequisite for being able to establish a proper infection. So this study, we collected European honeybees, obviously, to isolate sapwood virus. They were collected from Adelaide, and stingless bees were shipped over from Brisbane. Just a quick background on stingless bees. They're a tropical to subtropical social species with a eusocial structure similar to that of the European honeybee. They also have the close relationship to the European honeybee, being in the same family. And sapwood virus has previously been detected in foraging adults, making them a good candidate for this particular study. So we're guided by the research questions. Following our experimental inoculation, can sapwood virus replicate in stingless bee pupae? And does the virus have any effect on their survival? So I'll just quickly run through some of the methods. I think I need to speed this up a little bit. We collected symptomatic honeybee larvae that showed signs of sapwood virus. These were homogenized alongside a buffer to create a liquid, which was screened using PCR to confirm the presence of sapwood virus and the absence of any other contaminating viruses. Once this was confirmed to be capable of infecting honeybees, we could then proceed with this. We had stingless bee brood shipped over from Brisbane to Adelaide, and we screened them to confirm there were no viral infections already present. Then to investigate the replication ability of the virus, we injected pupae, the stingless bees, into the abdomen. They were then frozen either on the day of injection or a number of days post-injection to preserve the viral titer, the viral concentration at that point in time. These then had, once they were terminated, they had RNA extracted to isolate the virus, and they were then, the viral concentration was quantified using a method known as quantitative PCR. And here are the results of that experiment. Basically, pretty straightforward graph. On the y-axis, you have the virus concentration. And on the x-axis, you have the days post-injection. And each of the box plots indicates the distribution of concentrations in the bees from that treatment. So on day 0, you can see that there's really no difference between any of the treatments, the concentration. But when you get to day 3 and day 7, those blue-colored controls stay around the same level, whereas those with the sapwood virus, different levels of sapwood virus concentration, ranging from yellow up to red to indicate high concentration, they do increase significantly, showing that there is replication taking place in the bodies of those pupae. So we determined that the virus was able to replicate. We then wanted to determine whether this ability to replicate increased the risk, the mortality risk, or reduced survival in those bees. So a separate experiment inoculated the bees in the same way. They were then incubated at sort of normal temperatures that they would be in the nest, and daily monitoring took data on their development and the development of any symptoms. So some examples of different development stages can be seen on the left here, and development of symptoms on the right. So blackening of the body indicates that they're obviously not developing as they should be. And using this data, we determined a day of death based on some sort of halt in development or whether there was obvious sign of damage on the body. We then drew survival curves, which were analysed. The differences between them were analysed using a Cox Regression Analysis. And the results of that are shown on this graph here. Pretty straightforward. Again, on the y-axis is the proportion of surviving pupae over any given time on the x-axis. So importantly here, there's the medium concentration that we saw evidence of replication from was not significantly different from some of the controls, whereas that high concentration was significantly different from all the others, so it had a significantly reduced survival time. So while there doesn't seem to be a huge effect of the virus on survival time at quite a high dosage, there is an effect. So to just briefly conclude, this study was able to isolate sacbrood virus from symptomatic honeybee larvae and produce it into an inoculum. We were able to demonstrate that sacbrood virus is capable of replicating in stingless bee pupae, and that there is an increase in mortality in stingless bee pupae at that higher concentration that we used. And so briefly, what are the implications? We can determine that sacbrood virus is potentially harmful to wild stingless bee populations, and also managed stingless bee populations, of which there is a growing industry in the tropical eastern states. And we can also use these findings to better inform European honeybee management, particularly with the placement of European honeybee hives near areas of conservation interest with native bees in them. Not only do those native bees already have to deal with competition for floral resources with European honeybees, but it also seems like they might have to deal with the potential spread of disease from honeybees into those vulnerable populations. I think I'll just leave my future research and present my acknowledgement slide and just ask for any questions if we have time. And thank you all for listening.
Thanks a lot, Nick. Very, very interesting. And again, nice results. Very seems pretty clear cut that SBV is an issue. We've got time for one quick question.
Hi, Nick, it's Mary here. Can I ask you a question?
Yes,
I think you may have hinted at it with your future research, but can you explain why you chose to inject the pupae with the virus and how that, how that relates to how sacbrood virus is transmitted within honeybees?
Yeah, so that's a very good question. It's definitely the injected method is a much more artificial infection method than they definitely wouldn't be directly injected into the abdomen in the wild. So in the future, it would definitely be important to in some way feed the bees when they're larvae and determine what the effect of the virus is then. There are much more mechanisms in the gut to fight off infections like that, and we may see potentially less of an effect of the virus if it went through that sort of infection method.
Pretty good response there, Nick. Thank you. All right.